Welcome to the video lecture for Unit 5, Module 10.2.3. In this one, we're going to discuss the large intestines. As always, here are your learning objectives, so be sure to reference these when studying for both your lecture quiz and the unit exam. Let's go ahead and get started. The horseshoe-shaped large intestine begins at the end of the ileum and ends at the anus. The large intestine lies inferior to the stomach and the liver and almost completely frames the small intestine. The large intestine stores digestive waste and reabsorbs water. Resident bacteria in the large intestine are an important source of vitamins, especially vitamin K, biotin, and vitamin B5. The large intestine, also known as the large bowel, has an average length of about 1.5 meters and a width of about 7.5 centimeters. We can divide it into three parts. 1. The pouch-like cecum, the first portion of the large intestine. 2. The colon, the largest portion. And 3. The rectum, the last 15 centimeters of the large intestine and the end of the digestive tract. Materials arriving from the ileum first enters an expanded pouch called the cecum. The ileum attaches to the medial surface of the cecum and opens into the cecum at the ileocecal valve. The cecum collects and stores materials from the ileum and begins the process of compaction. The slender, hollow appendix, or variform appendix, is attached to the posteromedial surface of the cecum. The appendix is normally about 9 centimeters long, but its size and shape are quite variable. A small mesentery, called the mesoappendix, connects the appendix to the ileum and the cecum. Lymphoid nodules dominate the mucosa and submucosa of the appendix. The primary function of the appendix is as an organ of the lymphatic system. Inflammation of the appendix is known as appendicitis. The colon has a larger diameter and a thinner wall than the small intestine. Distinctive features of the colon include the following. The walls of the colon form a series of pouches, or haustra. The creases between the haustra affect the mucosal lining as well, producing a series of intestinal folds. Haustra permit the colon to expand and elongate. 3 separate longitudinal bands of smooth muscles, called the tennae coli, run along the outer surfaces of the colon just deep to the serosa. These bands correspond to the outer layer of the mucolaris externa and other portions of the digestive tract. Muscle tone within the tennae coli is what creates the haustra. The serosa of the colon contains numerous teardrop-shaped sacs of fat called omental appendices, also called fatty or epilotic appendages. We can subdivide the colon into 4 regions: the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. The first is the ascending colon, and it begins at the superior border of the cecum and ascends along the right lateral and posterior wall of the peritoneal cavity to the inferior surface of the liver. There, the colon bends sharply to the left at the right colic flexure or hepatic flexure. This bend marks the end of the ascending colon and the beginning of the transverse colon. Next is the transverse colon, and it curves interior to the right colic flexure and crosses the abdomen from right to left. The transverse colon is supported by the transverse mesocolon. It is separated from the anterior abdominal wall by the layers of the greater omentum. At the left side of the body, the transverse colon passes inferior to the greater curvature of the stomach. Near the spleen, the colon makes a 90-degree turn at the left colic flexure, or the splenic flexure, and becomes the descending colon. The descending colon proceeds inferiorly along the left side, reaching the iliac fossa, formed by the inner surface of the left ileum. The descending colon is retroperitoneal and firmly attached to the abdominal wall. At the iliac fossa, the descending colon curves at the sigmoid flexure and becomes the sigmoid colon. The sigmoid flexure is at the start of the sigmoid colon, an S-shaped segment that's only about 15 centimeters long. The sigmoid colon lies posterior to the urinary bladder, suspended from the sigmoid mesocolon. The sigmoid colon empties into the rectum. The large intestine receives blood from the branches of the superior mesenteric and inferior mesenteric arteries. The superior mesenteric and inferior mesenteric veins collect venous blood from the large intestine. The rectum forms the last 15 centimeters of the digestive tract. It's an expandable organ for the temporary storage of feces. The movement of fecal material into the rectum triggers the urge to defecate. The last portion of the rectum, the anal canal, contains small longitudinal folds called anal columns. The distal margins of these columns are joined by transverse folds that mark the boundary between the columnar epithelium of the proximal rectum and a stratified squamous epithelium like that in the oral cavity. The anus, or anal orifice, is the exit of the anal canal. There, the epidermis becomes keratinized and identical to the surface of the skin. The circular muscle layer of the mucolaris externa in this region forms the internal anal sphincter. The smooth muscle cells of the sphincter are not under voluntary control. The external anal sphincter, which guards the anus, consists of a ring of skeletal muscle fibers that encircles the distal portion of the anal canal. This sphincter consists of skeletal muscle and is under voluntary control. The lamina propria and submucosa of the anal canal contain a network of veins. If venous pressures there rise too high due to straining during defecation or pregnancy, the veins can become distended, producing hemorrhoids. The diameter of the colon is about 3 times that of the small intestine, but its wall is much thinner. The major characteristics of the colon are the lack of villi, the abundance of mucus cells, and the presence of distinctive intestinal glands. The glands in the large intestine are deeper than those of the small intestine and are dominated by mucus cells. The mucosa of the large intestine does not produce enzymes. Any digestion that occurs results from the enzymes induced in the small intestine or from bacterial action. The mucus provides lubrication as the fecal material becomes drier and more compact. Mucus is secreted as local stimuli, such as friction or exposure to harsh chemicals, trigger short reflexes involving local nerve plexuses. Large lymphoid nodules are scattered throughout the lambda and submucosa. The mucolaris externa of the large intestine is unusual because the longitudinal layer have been reduced to the muscular bands of the tendon coli. However, the mixing and propulsion contractions of the colon resemble those of the small intestine. Less than 10% of the nutrient absorption underway in the digestive tract occurs in the large intestine. Nevertheless, the absorptive operations in this segment of the digestive tract are important. The large intestine also prepares fecal material for defecation. The reabsorption of water is an important function of the large intestine. Roughly 1,500 milliliters of material enters the colon each day, but only about 200 milliliters of feces is ejected. To appreciate how efficient our digestion is, consider the average composition of feces: 75% water, 5% bacteria, and the rest is a mixture of indigestible materials, small quantities of inorganic matter, and the remains of epithelial cells. In addition to reabsorbing water, the large intestine absorbs a number of other substances that remain in the feces or were secreted into the digestive tract along its length. Examples include useful compounds such as bile salts and vitamins, organic waste products such as urobilinogen, and various toxins generated by bacterial action. Most of the bile salts entering the large intestine are promptly reabsorbed in the cecum and transported in blood to the liver for secretion into bile. Vitamins are organic molecules that are important as cofactors or coenzymes in many metabolic pathways. The normal bacterial residents of the colon generate 3 vitamins that supplement our diets. First is vitamin K, a fat-soluble vitamin in the liver that is required for synthesizing 4 clotting factors, including prothrombin. Intestinal bacteria produce about half of your daily vitamin K requirements. 2 is biotin, a water-soluble vitamin important in various reactions, notably those of glucose metabolism. 3 is vitamin B5, or pantothenic acid, a water-soluble vitamin required in the manufacture of steroid hormones and some neurotransmitters. Vitamin K deficiencies lead to impaired blood clotting. They result from either 1, not enough lipids in the diet, which impairs the absorption of all fat-soluble vitamins, or 2, problems affecting lipid processing and absorption, such as inadequate bile production or chronic diarrhea, frequent watery bowel movements. Disorders due to deficiencies of biotin or vitamin B5 are extremely rare after infancy. The intestinal bacteria generally produce sufficient amounts to supplement any dietary sources. We discussed the fate of bilirubin, a breakdown product of heme, earlier. In the large intestine, bacteria convert bilirubin to urobilinogens and stercobilinogens. Some urobilinogens are absorbed into the bloodstream and then excreted in urine. The urobilinogens and the stercobilinogens remaining within the colon are converted to urobilinogens and stercobilins by exposure to oxygen. These pigments in various proportions give feces a yellow-brown or brownish color. Bacterial action breaks down peptides that remain in the feces. The action generates 1. Ammonia in the form of soluble ammonium ions, indiol and scholate; 2. Nitrogen-containing compounds that are primarily responsible for the odor of feces; and 3. Hydrogen sulfide, a gas with a rotten egg odor. Significant amounts of ammonia and smaller amounts of other toxins cross the colloid epithelium and enter the hepatic portal circulation. The liver removes these toxins and converts them to relatively non-toxic compounds that can be released into the blood and excreted by the kidneys. Intestinal enzymes do not alter indigestible carbohydrates. These materials arrive in the colon virtually intact. These complex polysaccharides provide a reliable nutrient source for bacteria in the colon. The metabolic activities of these bacteria create small amounts of flatus, or intestinal gas. Foods with large amounts of indigestible carbohydrates, such as beans, stimulate bacterial gas production. Distension of the colon, cramps, and the frequent discharge of intestinal gases can result. The gastroileal and gastroenteric reflexes move materials into the cecum while you eat. Movement from the cecum to the transverse colon is very slow, allowing hours for water reabsorption to convert the already thick material into a sludgy paste. Peristaltic waves move material along the length of the colon. Segmentation movements, called haustral churning, mix the contents of adjacent haustra. Powerful peristaltic contractions, called mass movements, occur in a few times each day. They move material from the transverse colon to the rest of the large intestine. The stimulus is distension of the stomach and the duodenum. The commands are relayed over the intestinal nerve plexuses. The contraction force feces into the rectum and produce conscious urge to defecate. The rectal chamber is usually empty, except when a powerful peristaltic contraction forces feces out of the sigmoid colon. Distension of the rectal wall then triggers the defecation reflex. This reflex involves two positive feedback loops. Both loops are triggered by the stimulation of stretch receptors in the rectum. Rectal stretch receptors also trigger two reflexes important to the voluntary control of defecation. One is a long reflex mediated by parasympathetic innervation within the pelvic nerves. This reflex causes the internal anal sphincter to relax. The smooth muscle sphincter controls the movement of feces into the anal canal. The second is a somatic reflex that stimulates the immediate contraction of the external anal sphincter, a skeletal muscle. The pudendal nerves carry the modal commands. Both the internal and the external anal sphincters must relax for feces to be eliminated. However, the two reflexes just mentioned open the internal sphincter but close the external sphincter. The actual release of feces requires a conscious effort to open the external sphincter. In addition, other consciously directed activities can raise intra-abdominal pressures and help force fecal material out of the rectum. These activities include tensing the abdominal muscles or elevating intra-abdominal pressures by attempting to exhale forcibly with a closed glottis. If the external anal sphincter remains constricted, the peristaltic contractions cease. However, additional rectal expansion triggers further defecation reflexes. The urge to defecate usually develops when rectal pressure reaches about 15 millimeters per mercury. If this pressure is more than 55, the external anal sphincter involuntarily relaxes and defecation takes place. This mechanism brings about defecation in infants and in adults with severe spinal cord injuries.